Welcome to chapter 19, and in this chapter we are going to study about the cell cycle, DNA replication and mitosis. So the fundamental question is the why cells divide. You see, when the unicellular organisms or the multicellular organisms they grow and they have to divide in order to maintain their population, right? So whether the cells if they are single celled and whether if they are multicellular, they have to divide. In single cell organisms, population will be increased following the cell division, but in multicellular organisms both possibilities are there. Sometimes they will lead to the growth of the organism, and the second criteria is the recycling. You see, in the multicellular organisms like us, we have lots of cells that die during the course of their performance of their activities, and they expired and they are immediately replaced. So this is one of the reasons that why cells divide, because they have to grow and reproduce. So there are some examples in our body. For example, about 2 million stem cells in our bone marrow. They divide to maintain a constant supply of the red blood cells in our body. So it is a continuous process. Cell division is a continuous process in the multicellular organisms. No matter how old they are, the cell division, it keeps on and on to maintain the balance of the cells in our body till the time we are alive. And after the death, of course, there is no more cell division, and the body is completely destroyed, and it is then not subjected to any kind of cell division. So right from the beginning, like the fertilization until the end, the continuous mechanism, the regulation and the activity of the cell division, it is going on all the time. Okay, some of the cells they will divide more often than the other. For example, the nerve cells, or the muscle cells, they seldom divide, or sometimes we say that they never divide. But in case of the blood cells, they always divide in our bone marrow to keep the maintenance of the cells. And similarly, the cells that are responsible for our recycling of the lost tissues, organs of the cells, they will divide, and they will continue this property of cell division. Similarly, that I will give you one example of the liver. If we cut apart 30% of the human liver, the rest of the 70% is able to regenerate the missing 30%. This is called regeneration, and sometimes it is needed by the body, but not all the organs they can regenerate. Only some organs they have this capability of regeneration. So here you see an example: human embryonic and fetal development. You see the fertilized egg here, then we have 2 cell stage, then 4, 8, 16, then the blastocyst, and then we have fetus about of 20 weeks. And after 20 weeks there is an increase in the size, right? And it is believed that at 20 weeks the fetus is complete, and then the only difference in the later 16 months is that only the size of the cells they will increase. Okay, and so the size of the fetus will be increased, and after 36 months the baby is ready to go out in this world. So this blastocyst stage is very important. This is the stage where every cell within the blastocyst is capable of regenerated or produced as a single organism. For example, if you pick just one cell out of this blastocyst, this one cell will have complete mechanism, this cell will have complete information to be developed as a single individual. So for cloning purposes, scientists they took advantage of this stage, and during the blastocyst stage they just pick 1, 2, 3, 4, 5, 6, 7, 8, whatever they need, and they grow it in the lab, and they are most of the time they are successful, and they have done experiments with the animals and they have been successful in that, especially for the cloning purposes and for other purposes. In Korea, it is legal to have a clone of your pet, for example cat or dog. They have average life of 10 years or 15 years. So what people do, they just go to that center and they book a clone for their pet. So in the next slide here, you see the prokaryotic cell cycle is very simple. This is a process called as binary fission. Just one cell, mother cell, it is divided into two daughter cells. Okay, and but two things are necessary for their cell cycle or for their cell division. Those two things are, one is food. There must be enough food, and the second thing, there must be enough space. The E. coli, the simplest of the prokaryote, like the E. coli, it will just take 20 minutes to double its population, provided they have enough food with them and enough space. But the eukaryotic cell cycle is somehow different from the binary fission and from the rest of the prokaryotes. It has M phase. M stands for mitosis. This is the mitotic phase, and then the green one, all that green arrow, it shows interphase. So there are two phases. One is the M phase and the other one is the interphase. So most of the time the cell is in the interphase. Interphase is a kind of phase where cell is not dividing. Okay, M is the phase where cell needs to divide, and this cell will be converted into two different cells, two daughter cells, of course. Okay, if we dissect M phase, so M phase consists of the mitosis and cytokinesis. Now mitosis is the cell is going to be divided into two different daughter cells. One is the first stage will be the chromosomes, they will be condensed, and then we have sister chromatids. You see the prophase, metaphase, anaphase, telophase, and after that the cytoplasm of the two cells, of the two daughter cells, that will be separated from each other, and they will give rise to two different complete individual cells. Okay, so these are the phases. We will see how that M phase is regulated. There are different checkpoints. Checkpoints is a place, is a location where it is checked whether cell has enough amount of nutrients, enough amount of food, enough amount of the signals that it should be proceeded for the division. Okay, so we'll see in the next slide that how these stages, what are the steps of these different stages. So here we have phases of cell cycle. M phase, it is very, very short, just 1 hour. So most of the time cell will be in the interphase, and the S phase is called as a synthesis phase. This is the synthesis of what? Synthesis of DNA. Okay, the nuclear DNA, it becomes photocopied. We have two copies, complete copies of the DNA during the S phase. So during the G1 phase, that separates S phase from the preceding M phase, a second gap that called as the G2 phase, it separates the end of the S phase from the onset of the next M phase. So it is very important. These gaps are very important, and in between these gaps there are checkpoints who will confirm, who will ensure that the cell is going towards the right direction if he or she is going to divide. S and M phase lengths, they are different for different mammalian cells. Sometimes the cells, they remain in the G1 phase for 8 to 10 hours, but the major decision whether the cell has to divide again, it will come during the G1. So the cells that are present awaiting a signal that they should divide or not, they will always be in the G1 phase. Okay, and they are going to trigger into the cell cycle, and then commitment to divide, they are said to be in the G0 phase. So there is no signal. If there is no signal for the cells to divide, they will always be called as the G0. So they are never going to divide. especially the nerve cells, they are always. Flow cytometry. Flow means to move, and cyto means cell, metry means to measure. So these three words, they are a definition in these. The meaning of the flow cytometry is within these words. Okay, so this is a technique that is used to measure the cells when they are moving from one direction to another. So this automated analysis, it is capable of the analysis of millions of individual cells simultaneously. So the first step will be that they will be stained with one or more fluorescent dyes, and we have different dyes for the DNA, we have different dyes for the proteins. Okay, and these dyes are added in the mixture of cells. The cells are then passed through a liquid stream through the beam of laser light. Okay, the cells will be passing and the beam of laser light will be crossing them. So by analyzing the fluctuations in the intensity and colors of the fluorescent light that is emitted by each cell as it passes through the laser beam, it will be captured. When it is captured, it is measured, and there will be a specific output, and based on that output, scientists will decide that this cell is actually belongs to this phase, or this cell is actually belongs to this particular tissue, or something like that. So because when the cell is dividing, there will be huge DNA over there. Okay, as I told you that previously, that when the cell needs to divide, it is in the S phase. So during the S phase, the copies of the DNA are doubled. So these double copies will be stained, and they will give double fluorescence. And they will emit double fluorescence, and this double fluorescence will let us know that this cell is actually going to divide. So it will help the physicians to decide that if they see lots of such cells where the DNA is doubled, so they can make a decision that there is something wrong with these cells. They are going to grow into cancerous cells. So flow cytometry is very, very important as a diagnostic tool for the cancer cells. Here is the principle for the flow cytometry. Here you see that we will add a fluid containing cells in the container, and that container will just make sure that cells will pass through a tiny hole one by one. Okay, just in a row. So every cell then comes in contact with this laser. So this laser will capture the images and the inside, the materials inside, and depending upon the fluorescent material emitted from the stained cells, it can be decided that the cells are having double DNA, or cells are having less DNA, or the cells have no DNA. So the mathematical models are available for that, and immediately we can go for the diagnosis that what type of cells are there, whether the cells are in the division phase, whether the cells are not dividing, something like that. Okay, so flow cytometry is very important in the diagnosis of the cancers as well. So in the Ashok Khanna Memorial Hospital, this facility is available, and most of the diagnosis is through flow cytometry. So the next topic is DNA replication. As you see, replica means copy, and when we are going to make copies of the DNA, that process is known as DNA replication. And DNA replication, it occurs during the S phase, right? Remember S phase that comes after the G1 phase. G1 phase, then we have S phase, then we have G2 phase, and then we have finally the M phase. M phase consists of the mitosis and the cytokinesis. So DNA replication is semi-conservative, as all you know. Semi-conservative means that one of the strand of the daughter cell's DNA will always be from the parent strands. Okay, here you see there are two strands. One will go towards one daughter cells, and the other will go towards the other daughter cells. So that's why we call it as a semi-conservative method of DNA replication, and it was confirmed through this classical experiment in which the N15 and the N14, two different isotopes, they were used for the growth of the bacteria, because we know that if during DNA replication there is a need of the nitrogen, right? So if the more nitrogen is used, it means that more signal will be produced and more heavier will be the band after the centrifugation. Okay, so this classical experiment shows us that when the N15 and N14, both of them, they are present in the next generation of the E. coli cells. So these E. coli cells, they confirmed the first generation of DNA. They were having the semi-conservative methods, and then in the second generation we have semi-conservative, as well as we also have the original DNA, and we also have the daughter's DNA that were totally new. Okay, that shows that if we are having one original DNA, it will always give rise to one original DNA strand and one new daughter strand that will be replicated through this original strand. Okay, so that's why we call it as semi-conservative replication. So DNA replication has very few steps here. Helicase, that is going to unwind the helix and separates the strands, and primase, it anneals the RNA primers. Then we have polymerase enzyme, it copies each strand, okay, continuously on the leading strand, as well as the Okazaki fragments on the lagging strands. The polymerase one, it replaces the primers with the DNA nucleotides, and then the ligase enzyme, it seals everything up. Okay, so this process looks very simple in this slide, but actually it is not very simple. It has lots of other proteins, lots of other enzymes responsible for, and I will share some animation of this DNA replication with you guys, and I hope you will like that also for your understanding. So all these processes will end up in the formation of two identical copies of the original DNA molecule. So this is the origin of replication here. This is the example from a bacterial cell. Here you see the bacterial DNA. This bacterial DNA is trying to replicate its DNA, and from one copy they are getting another copy, and this is just like a double spring. When the springs are overlapped on each other and we just remove one spring from the other one, it's just like that. Here you see. Okay, so replication fork is their origin of replication. Origin of replication is the point where actual replication is going to start. Okay, we have lots of origin of replications in the eukaryotic cells, and I will ask you why there are so many origin of replications present in the eukaryotic cells. Okay, and I will ask this question in our assignment. So these origin of replications, the replication is started, and after replication forks, they move in the opposite directions, and after that they are separated from each other through the process of the replication, and the circular DNA replication is ended up like that, and we have two daughter nucleic acids. Okay, and after that the cytokinesis process, where the cytoplasm of the cells, of the bacterial cells, it is divided, and here we got the two daughter cells from one single bacterial cell. These are some of the descriptions of the daughter strands and the creation of the replication bubble. This is the replication bubble. A replication bubble is a place where origin of replication has just started. Okay, so two replications on the left fuse together, and replicons, they fuse together, and then they are going to give rise a complete daughter chromosomes here. Okay, in the eukaryotic cells as well as in the prokaryotic cells, there are number of origin of replications on the DNA. Okay, but the number of origin of replications, we say them ORI, or this origin of replication are much more in the eukaryotic cells, and they are also present in the prokaryotic cells, but in less numbers. So what is there, that immediately that origin of replication they start, and we have lots of bubbles at the same times, and in a minimum period we get lots of bubbles, and those bubbles are then fused together so that we can have a full length DNA copy in a shortest possible time. That's the reason there are so many. So here we are seeing the image of cell cycle, G1 S phase and G2 phase. Here see the dotted lines. These dotted lines are saying that we need some kind of licensing system. You see, this is the replication of DNA. It is not an easy job, and it is also not very common. That without any purpose, DNA is not going to replicate, and without the real need, cell is not going to divide. And if there is no need by the organism of the divided cell, then it will be a problem. You see, in all the cancers, what happens? The cells are not needed to divide, but they divide. That's why it is called as cancer. And that division of the cell, it is not controlled. This type of cell division we can control it, because if we don't have any requirement of the cell to divide, then we can stop it at some point. So that's the whole concept here. So the CDK and gymin, these are the proteins that are going to inhibit the licensing system. When they are going to inhibit the licensing system, when the cell is not required to divide, they will inhibit it. But when the cell is needed to divide, the licensing system will become active, and it is composed of different proteins, helicase loaders, ORC proteins, MCM proteins, and pre-replication complex. All that licensing system is then going to activate the S phase, and this S phase is going to cause the division of the cells later on. These are different enzymes that are responsible for to carry out the replication in bacteria and also in the eukaryotes. So there are lots of different enzymes. This is not very simple step. It is very complex procedure within the cell, and we have lots of different enzymes responsible. I'm not going to ask you the function of each and every enzyme, but I would like to understand, for example, what is the function of the primase. Primase means that they are going to add the primer. Okay, so primer will be synthesis. And why the primers? They are always RNA. Okay, this is again a question I am leaving for you. The answer I will ask you. I will seek answers from you about the question that why the primers, they are of RNA origin and not of the DNA origin. Okay, so what is the answer to that? So we have lots of different initiator proteins. Ligase is going to seal up the gaps through covalent bonds formation. Okay, so these are different enzymes, and this is the concept of growing chain, growing in a strand. And as we know that adenine has a double bond formation with a thymine, and guanine has a triple bond formation with a cytosine. And these nitrogenous bases, they have different hydrogen bonds, and then we have double-stranded DNA, and this is the incoming DNTP, okay, deoxynucleoside triphosphate, and this nucleoside is going to make a double bond with the nitrogenous base of the template strand, and one of the phosphate, the pyrophosphate, will be removed during this process. Okay, so this is a 5' end, and this is a 3' end, and this is a 5' end, this is a 3' end. It means that the template strand and the growing strand, they are anti-parallel to each other. They are not in the same direction. And again, this 5' to 3' direction of the ribose sugar here, deoxyribose sugar here, it is going to provide us more stability when they are in anti-parallel direction. It gives more strength to the DNA strand. So here complete DNA is made, you see, and the formation is always from the 5' to the 3' direction. Here you see the 5' direction is here, and then 3' direction is here. So the nucleotides will always be added here. Here you see the new phosphodiester bond is made, and here this bond is made. These bonds are already made up here in the template strand, but the nucleotides can only be added at the 3' end only, not at the 5' end. Why? Because we have a 3' hydroxyl group here, and this group can be manipulated to create a new phosphodiester bond, and that's why the direction of DNA synthesis is always from the 5' direction to the 3' direction. Here you see the replication fork. This is the leading strand, this is the lagging strand. On lagging strand again the formation is also from the 5' to 3', and we will have a separate RNA primer for each Okazaki fragment of the lagging strand, and leading strand we just need one primer and that's it. And this overall direction you see it is from the 5' to the 3' end. Again it is from the 5' to the 3' end. Okay, so the newly synthesized DNA will always be made. Look at the heads, look at the arrowheads where they are moving from 5' to 3', from 5' to 3', from 5' to 3'. So the direction of the synthesis of the new DNA it is always from the 5' to the 3', and the reason I have told you that at the 3' end we have a free hydroxyl group. So what happens? Proofreading. Proofreading means the exonuclease activity. Sometimes during the process of photocopying of DNA, like the replication of DNA, misincorporations of the nucleotides, they are likely to occur because this is a very fast process, and sometimes the incorrect base may be added. For example, T always binds with A, but sometimes if T binds with G, or if T binds with C, or if A binds with G or with C, this is called as the incorrect base incorporation. So this must be removed. And how they are removed? They are removed through proofreading. What is proofreading? This is the activity of the DNA replication enzyme, DNA polymerase, that it can sense that I have done an error, some misincorporation has been done, so I have to go back and repair it. So what will happen, that the polymerase enzyme will come back, and exonuclease activity of the polymerase removes the incorrect base and then add the correct base on it, and then moves further to complete the process of replication. But sometimes these errors are also not recognized by the polymerase, but still nature has some DNA repair mechanisms, DNA repair enzymes that perform this function. We will see that how those DNA repair systems they do work. Okay, so primase, they are going to add the primer. Here the RNA primer is synthesized by the primase enzyme. This RNA primer is synthesized, and newly made DNA is from the 5' to 3' directions. Okay, so here the nucleotides are added one by one. DNA polymerase 3 uses the polymers to initiate the DNA synthesis from the 5' to 3' direction, and after that the DNA polymerase 1 replaces the DNA with the DNA here, and ligase enzyme they are going to seal the gap. Okay, this is going to be done in no time. In just milliseconds this replication process is completed. So the initiator here, this image is showing the initiator protein is binding to the double-stranded DNA at the origin of replication, and of course ATP is involved here. So after the replication fork is made, DNA helicase has performed its function. Single-stranded binding proteins here, they keep the strand, both strands as single strands, because we need both strands as single strands so that they can be polymerized through polymerase enzyme. So DNA guidase, this is the topoisomerase enzymes. It will release the supercoiling, and then we have the formation of the primer through the primase enzyme. And after that, after the primase enzyme have given the primer, then the leading strand will go forward towards the 5' to 3' direction, and in the lagging strand it will also go towards the 5' to 3' directions. Okay, so DNA polymerase 1 is the enzyme that will remove the primer. Okay, and after removing of the primer, the gaps are filled by the DNA ligase here in the seventh steps. And after the DNA ligase has filled the gaps, what will happen? The complete replication of the DNA is there. And then in the next video you will see the cell aging. You see that this is funny, this is old DNA, this is a newly synthesized DNA. So we will discuss some interesting facts about these. So cells do grow old, right, like us. So why? Because we in our cells, you see at the ends we have telomeres. Telomeres are the at the time of birth, their length is about 10,000 base pairs. These are the short tandem repeats, okay, and these repeats are going to be less and less and less as the cells grow and grow and grow and divide and divide and divide. So whenever the cell divides, this telomere is shortened. Because of the shortness of the telomeres, the appearance of the properties of the old people, like the wrinkles on the face and weaknesses, they appear. Why? Because the most of the functional things, the genes that are responsible for the functions of the bodily activities, they are located over here. So when there are no telomeres, because telomeres do not encode any protein, so when they are not present anymore, the genes over here, they are subjected to the exonuclease activity, and when they are subjected to exonuclease activity, it means that their function is destroyed. So that's why the telomeres, they are so important in our cell division. Okay, so here is the process. I will also share a separate video for that how telomeres, they are important, and how telomeres, they are also sometimes bad for the research, because some people are working on the anti-aging things, that how the human beings can be immortal, something like that. Okay, so here you see here the telomeres, they are present on the ends. Here you see 5' to 3', and here 5' to 3'. Here you see you cannot add primer over here anymore. So that's why the nature has given a telomerase enzyme. Telomerase enzymes are present in our nucleus, and in that nucleus they will repair the end, and they have their own primer, they have their own nucleus, they have their own enzyme, the polymerase enzyme that is known as telomerase, and this enzyme will incorporate, it elongates this 5' to 3' direction, so that a new primer can bind here, and they can further go up to repair or to the repeats, or to save the telomere repeats. Okay, so that's why. But when cells divide again and again, so this portion of the DNA, it is subjected to the destruction of the exonuclease activity, and this portion is encoding the proteins, and when these proteins are not encoded properly, then we feel the properties of aging. Okay, so here you see the primer is added, and then again we are going to have a new strand somehow, but again you see the portion is there. This is the shortening of the telomere. You cannot add any more primer anymore here. So if there is no primer, then it will on every cycle it will become shorter and shorter and shorter. So the Hayflick limit is there. What is the Hayflick limit? It is the limit, the number of times a human cell or the eukaryotic cell or the mammalian cell can divide, and that Hayflick limit is 50. Our cell cannot divide more than 50 times in our average life. So that's why before the whole Hayflick limit, the telomeres, they are shortened and shortened, and as a result, whole the DNA is subjected to the destruction, and then following the process of aging, and then ultimately death welcomes us.